Hi dear students. Welcome to our session on growth curve and its significance. Living organisms grow and reproduce. When microbes are provided with nutrients and appropriate environmental factors, they become metabolically active and grow. Growth common refers to increase in a microbial size or population number or the both. Growth also results when cells simply become longer and larger. It involves an increase in the number of cells rather than in the size of individual cells. The change in population in bacteria chiefly involves binary fission. Growth is defined as an increase in cellular constituents which leads to a rise in cell number. Microorganisms reproduce by binary fission or budding. The cells copy their DNA almost continuously and divide again and again by the process called binary fission. Binary fission is a form of asexual reproduction in single-celled organisms by which one cell divides into two cells of the same size. There are two levels of growth. The first one is that a cell synthesizes new components and it increases its size. The second level of growth is the increase in number of cells in the population. The bacteria divide by binary fission. When a bacterial cell reaches a certain size, it divides to form two daughter cells. The nuclear division precedes the cell division and therefore, in a growing population, many cells carrying two nuclear bodies can be seen. The cell divides by a constructive or pinching process or by the ingrowth of a transverse septum across the cell. Let's see the stages of binary fission once again. A parent cell prepares for division by enlarging its cell wall, cell membrane and overall volume. Midway in the cell wall develops notches like this that will eventually form the transverse septum and the duplicated chromosome becomes affixed to a special membrane site. Here you can see the cell wall in brown color and the cell membrane in a light yellow color. And in the next stage, the septum wall will grow inward and the chromosomes are pulled toward the opposite cells and as the membrane enlarges, other cytoplasmic components are distributed to the two developing cells equally. The septum is synthesized. In the next step, the septum is synthesized completely through the cell center and cell membrane patches itself so that there are two separate cell chambers like this. And at this point, the daughter cells are divided. Some species will separate completely, as you can see here, while the others will remain attached, forming chains or doublets. Doublets. The interval of time between two cell divisions or the time required for a bacterium to give rise to two daughter cells under optimum condition is known as the generation time or population doubling time. The process of cell division is a continuous process, as you can see. One cell will be giving rise to two cells will be giving rise to four cells and it will be going so on. Now let's see the examples of some organisms generation time. In coliform bacilli now let's see few examples. In coliform bacilli, that is E. coli and other medically important bacteria, the generation time is about 20 minutes. And in Staphylococcus aureus, it's around 27 to 30 minutes. In Bacillus subtilis, it's between 25 to 27 minutes. Some bacteria are slow growing and the generation time of Mycobacterium tuberculosis is 18 hours and in leprae it's 14 days and Treponema pallidum it's 1980 minutes. Now let's take E. coli as an example. E. coli is generation time is 20 minutes in optimal conditions. So after 20 generations, there will be 1 million cells and this will be taking around 7 hours. And after 30 generations, there will be 1 billion cells and this will be taking around 10 hours. Growth curve. The increase in cell number or growth in population is studied by analyzing the growth curve of a microbial culture. Bacteria can be grown or cultivated in a liquid medium in a closed system or also called as a batch culture. In this method, no fresh medium is added and hence with time, the nutrient concentrations will be getting depleted and an increase in waste is observed. As bacteria reproduce by binary fission, the growth can be plotted as the logarithm of the number of viable cells versus the time of incubation. The viable cells measures the number of living cells, that is, the cells that is capable of multiplication. When a bacterium is seeded into a suitable liquid medium and incubated, its growth follows a definite course. Bacterial counts are made at intervals after inoculation and plotted in relation to time. A growth curve is obtained and the curve shows the 4 basic phases of growth. First one is the lag phase, second one is the log or exponential phase, third one is the stationary phase and fourth one is the death phase. So here you can see the bacterial growth curve with the lag phase, log phase, stationary phase and the death phase. In the x-axis it is plotted with the logarithm of number of bacteria and in the y-axis the time in hour. Now let's see each phases in detail. The first one is the lag phase. It is the phase that is immediately following the seeding of a culture medium or as the cells are introduced into the new medium, no immediate increase in cell number occurs, though there may be an increase in the size of the cells. During this phase, the cells are undergoing a period of intense metabolic activity involving synthesis of enzymes and various other molecules required to divide in the coming phase. This phase can vary considerably in length depending on the nature of the medium and the microorganism. The medium may be different from the one in which the organism was growing previously. Here, the cells may be old and depleted of ATP, and essential cofactors and ribosomes must be synthesized before the growth can begin. So the microorganism requires times to recover and young and vigorously growing cultures and fresh medium are to be used for the lag phase to be short. The duration of the lag phase varies with the species, size of inoculum, nature of culture medium and environmental factors such as temperature. So in the growth curve you can see that the lag phase the number of bacteria is significantly low. The next phase is the log phase. In this phase, the cell starts dividing in a logarithmic way and this is also called as exponential phase. And the growth in this phase is balanced. Cellular reproduction is high during this period and the flow during this phase is a straight line. The cells are most active metabolically and the population is most uniform during this phase. Hence, exponential phase cultures are usually used in biochemical and physiological studies. The cells are growing and dividing and increasing in cell number. The rate of exponential growth of a bacterial culture is expressed as generation time or doubling time of the bacterial population. So, the generation time is defined as the time per generation where n is the number of generations. Hence, G is equal to T by N is the equation from which calculations of generation time can be derived. Exponential phase or log phase is balanced growth. That is, all the cellular constituents are manufactured at constant rates relative to each other. If nutrient levels or other environmental conditions change, unbalanced growth will be resulting. Organisms in a tube of culture media can maintain logarithmic growth for only a limited period of time, because as the number of organisms increases, the nutrients are used up, metabolic waste gets accumulated, and the living space may become limiting factor, and the aerobic organisms suffer from oxygen depletion. So in the graph you can see that if the logarithm of the viable count is plotted against time, a straight line will be obtained since the cells start dividing and their numbers increase exponentially. Another important significance of the logarithmic phase is that the primary metabolites are formed in the logarithmic phase or log phase, or to be more specific, the trophophase of log phase. The primary metabolites are involved in growth, development, and reproduction of the organism. It is typically a key component in maintaining the normal physiological process. Thus, it is often referred as central metabolite. They are typically formed during the growth phase as a result of energy metabolism and are deemed essential for proper growth. Examples of primary metabolites include alcohol such as ethanol, lactic acid, and certain amino acids. They are formed when all the nutrients are provided in the medium, and it's essential for survival and existence of the organism and its reproduction. Here you can see the example of exponential growth. You can see the time, the number of division, and the total population at the end of the division. The next phase is the stationary phase. Reproduction rate is balanced by the death rate and population remains constant here. There is exhaustion of some nutrients and accumulation of some toxic materials. In this stage, microbial death is equal to the microbial growth, and there is a period of equilibrium. The metabolic activity of surviving cells slows down, which stabilizes the population. The exponential growth cannot be continued forever in a batch culture. For example, it's a closed system such as a test tube or flask. The population growth is limited by one of the three factors in stationary phase. The first factor is the exhaustion of available nutrients. Second one is the accumulation of inhibitory metabolites or end products. And the third is the exhaustion of space. And in this case, it's called a lack of biological space. The significance of the stationary phase is that the secondary metabolites are formed in this phase. The secondary metabolites do not play a role in growth, development, and reproduction like primary metabolites do. Many of the secondary metabolites have a role in ecological function, including defense mechanism. For example, antibiotics and the production of pigments. Examples of secondary metabolites include erythromycin, bacitracin, and a component called atropine, which is derived from the plants. They are formed in the idiophase of the stationary phase, and they are produced in abundance, and hence they are industrially important. So when you look at the growth curve, you can see that the viable count remains stationary as an equilibrium exists between the dying cells and the newly formed cells in the stationary phase. And the next phase is the death phase. And in the graph itself, you can see a phase of decline in the death phase. In death phase, there is a decline in the number of viable cells. This phase also is like the log phase, but the cell number is declining in a logarithmic way. The cell is said to be dead if it does not revive itself and reproduce when incubated again in a fresh medium. In this phase, the number of live cells decreases at a logarithmic rate, as indicated by the straight downward sloping diagonal line in the growth curve. The duration of this phase is as highly variable as the duration of log phase. Both depend primarily on the genetic characteristics of the organism. Let's see few of the characteristics of the death phase. There is nutrient deprivation, waste material accumulation, irreversible loss of the ability to reproduce, and the death can be logarithmic and can vary with environmental conditions such as the growth medium employed and the microorganisms involved. An exception is that the resistant bacteria show extended survival. So let's once again sum up the phases of the bacterial growth curve. The first one is a lag phase. Here you can see there is no increasing number of living bacterial cells. And then the logarithmic phase or the exponential phase. Here there is exponential increasing number of living bacterial cells. And then it is followed by the stationary phase. In this, it's a plateauing number of living bacterial cells, and the rate of cell division and death are roughly equal, and it is followed by the death or the decline phase. In this phase there is exponential decrease in number of living bacterial cells. Let's see the significance of the bacterial growth curve. The study of bacterial growth curve is important when aiming to utilize or inoculate no number of the bacterial isolate. For example, to enhance the plant growth, to increase biodegradation of toxic organics, or produce antibiotics or other natural products at an industrial scale, and also the knowledge of bacterial growth kinetics and bacterial numbers in a culture medium is important from both a research and commercial point of view. Growth kinetics is also useful for assessing whether a particular strain of bacteria are adapted to metabolize certain substrates such as the industrial waste or oil pollution. Bacteria that are genetically engineered to clean up the oil spills, for example, can be grown in the presence of complex hydrocarbons to ensure that their growth would not be repressed by the toxic effects of oil. So for all such studies, we need to know the pattern of growth of each microorganism and their respective generation times. Similarly, the slope and shape of growth curves produced from bacteria grown with mixtures of industrial waste products can inform scientists whether the bacteria can metabolize that particular substance and how many potential energy sources for the bacterium can be found in the waste mixture. We have already discussed about the generation time in the exponential phase, and it is defined as the interval of time between two cell divisions or the time required for a bacterium to give rise to two daughter cells under optimal conditions. The generation time for bacteria vary from about 12 minutes to 24 hours or even more. The generation times for some common bacteria under optimal conditions of growth are given. For E. coli, if the medium contains glucose and salt, the generation time will be around 17 minutes. Bacillus megaterium, it's 25 minutes. Streptococcus lactis in the presence of milk, it's 26 minutes. Streptococcus lactis in the presence of lactose growth, it's 48 minutes. And then Lactobacillus acidophilus, it's 66 to 87 minutes. In Rhizobium, it's around 344 to 461 minutes. Even it is used in the medium of mannitol salt or yeast extract. And for Mycobacterium tuberculosis, when synthetic mediums are used, the minutes are 792 to 932 minutes. And in Treponema pallidum, when it is cultivated in rabbit testis, it's around 1980 minutes. Let's have a look on the calculation of generation time. When growing exponentially by binary fission, the increase in a bacterial population is by geometric progression. If you start with one cell, when it divides, there are 2 cells in the first generation, 4 cells in the second generation, 8 cells in the third generation, and it goes on. The generation time is the time interval required for the cells to divide. So it can be expressed as G is equal to T by N, where G is the generation time and T is the time intervals in hours or minutes. And also there are constants B, capital B and small letter b, where capital B is the number of bacteria at the beginning of a time interval and small letter b is the number of bacteria at the end of time interval. And N is the number of generations, that is the number of times the cell population doubles during the time interval. and it can be expressed as b, small letter b is equal to capital B into 10 raised to the power n, and this equation is an expression of growth by binary fission. You can also solve for n and g separately using the basic equations. So it can be expressed as log b is equal to log b plus n log 2, and after substitutions we can get that n value as 3.3 log b by b, and that value of n that we got in the previous session can be used to solve for g,